we explain why and how immortal cells are distinct from primary cells. Immortal cells have been a topic we come back to repeatedly, as they seem to be the way of the future when we are talking about emerging biotechnology, reagents, and equipment. Immortalized cells are used as a mainstay of this industry. The reason for this is simply that primary cells are unable to achieve the same results. Yet despite that, they have their place. Our recent videos on a permanent supply of red blood cells that are universally useful, organs on a chip, and more all relied on cultured cells. If these cells were made from primary cells, then they would not be possible. Immortalized cells had an opportunity to shine because of this. The immortal cells can be cultured continuously. This means there is no loss of function, gross mutations, or more that alter and ultimately act detrimentally to the cell's form and function. This is why we went over the procedure and existence of immortalized cells previously, the short version of which is that a change is made to the genome of the immortalized cells that allow them to be eternally copied. This is true no matter how many times the cells divide, which is very much like a tumor.
Sorry, kid.
That's alright.
A nice baby.
I'm not a baby, I'm a tumor.
What separates a tumor from a normal cell is the continuous and uncontrolled cell division. It could also be a loss of function and others, but for this video, let's focus on a useful element. This occurs due to the loss of various genes like the p53 suppressor gene. This can occur due to mutation or damage. This gene and others are active when a cell gets too old, begins to behave abnormally and more. What we are looking at is what defines a cell's age. Unlike a complex whole being, it is not measured in days, weeks, months, and years. Nor does it use any other standard measure of time, but an internal clock known as telomeres. These are nucleotide sequences at the end of the DNA that make a chromosome. Over time, this strand gets shorter, which is like the ribbon of your life slowly being used up. It can be expedited by stress, damage, and exposure to toxic material. This makes the telomere shorter, and as it gets shorter, the chromosome, and consequentially the genome within it, loses stability. That makes the cell more prone to mutations. This is a distinct feature of primary cells. Primary cells age where immortalized cells do not. Since primary cells age and immortalized cells do not, you can think of them as being your cells. They are the normal cells of a living being, the cells that age and die only to be replaced. This is exactly what you get when harvesting primary cells. Harvested primary cells are a powerful research tool when looking at diseases, and especially human diseases. This could be cancer, diabetes, cardiovascular disease, any number of neurological diseases, and so on. They are the cells of this pathology taken from the host. They contain the real, original genomic form of a current case. These cells have undergone, by definition, almost no changes. They haven't been divided and replicated continuously like a research sample would. This means that they are representative of the total and primary functionality of the tissue and pathology in which you are investigating. If not looking at a pathology, but instead the normal function of a tissue or cell type, you can also garner this information. This lets you have a copy of the disease or the cell from which to draw data. These are also cells that undergo the normal process of the cell cycle and ultimately cell death. This element gives rise to considerations in primary cell research that do not apply to immortalized cells, the premier concern being viability. Viability is a product of the cell's health and the number of passages that have occurred. Let's look at passaging before coming back to viability. A passage is the process of subculturing your cell sample. That is the culturing and therefore multiplication of the cells after the original sample is taken. Think of it as being like a sourdough yeast culture. Every 12 hours, yeast divides, and so you divide it and feed it to keep the cells in a good, healthy environment, nourished and removing toxic material. Despite your best efforts, over time this yeast begins to deviate from the original, if only by a little. In a similar manner, primary cells can be cultured, but as they are cultured, you need to provide them with new nutrition, take away waste material, and others things that would occur naturally in the body but must be done by hand in a lab. Over time, the stress of the culturing process leads them to develop mutations, little bits at a time, but these mutations add up in the long term. This is why most labs set a boundary on the number of passages, these often being 3 stock tube passages and then 3 working passages. When you include the original stock from the supplier, this makes for 7 passages in total. This is an important number to remember when you are sourcing cells from a reference laboratory or receiving cells from another laboratory. They will have already been passaged. For cell samples collected from novel sources, this is still important to remember for the cell quality. Passages directly relate to the reliability of the research coming out of the sampled cells. The more passages, the more deviation from the original stock, the less reliable the results are. Now for viability. It's important to try and remember that cells need to be able to perform their original function, whether they are immortalized or primary. You are going to have a better chance of getting an accurate picture of the viability of primary cells than you are immortalized cells if you're looking at a relatively normal cell function. These cells will have been derived and then isolated after a period of senescence. That is when they stop dividing, and that needs to be kick-started again. This is often done using the appropriate growing media. Then you add into this a number of growth factors and other agents, let's say, that will encourage it to perform as desired. These could be as simple as nutrients, but might also be modifications to the gas and pH. The viability is then measured by a number of things, the first being cellular confluence. That is, how well the cells begin to inhabit the area that they've been cultured into. Most cell culture is done in a flask or similar vessel. The surface of the flask will be at the bottom, above this will be the cells, and then above this should be the growing media. This is the media which is feeding it and promoting its growth. In an ideal world, you would get maximum confluence, meaning 100% of the surface would be completely covered in cells. The various figures that come up will be based on the viability of the cells, and the decreasing viability means you will get decreasing confluence. As little as half of your cells might prove viable in that respect. It's also a means of tracking and, to a certain degree, assessing how healthy your cells are, and if you should have called a stop to the culturing of the cells at a prior passage. It also tells you when you need to look at subculturing it to go on to the next passage. If there aren't enough cells binding to the surface, then you should look at subculturing, or whether or not the cells are unhealthy. This is where another element comes in, and that is cell counting. In order to get an idea of how many cells there are within a cell sample, you need something called a hemocytometer. These are normally combined with your cells and a dye called trypan blue. The hemocytometer is a specialized piece of glassware coming in the form of a very thick glass microscope slide. It has regular markings on it that create a chamber. You combine the chamber, the markings, and the size of your cell to figure out how many cells are in a given volume of media. You should now have an idea of how healthy the cells are and how many cells you have from your growing media. After this, you then need to look at cryopreservation, and this is the preservation of the primary living cells. This is done by combining the cells that are being grown with a variety of specialized medias. The specialized media allows you to freeze the cells without developing ice crystals inside the cell, destroying them. This is normally done by freezing the cells in a slow process at approximately the loss of 1 degree centigrade per minute in combination with the media used to freeze it. These can then be kept at a frozen state of less than 130 degrees centigrade. The freezing and unfreezing of these samples does not count as a passage, and so when you create your first 3 culturing passages, you produce more cells than you can use immediately, and you can then have 3 more passages after that to create even more cells. This means a small sample can create a large amount of working material at the end. The key takeaway here is that primary cells are a finite sample of the original host. They take a snapshot of the organism's state, but one that can only be tested so far and for so long. It does have the advantage of being an accurate, if not precise, measure of the elements being investigated. Unlike immortalized cells, you cannot continue to propagate them. You cannot necessarily easily genetically modify them or otherwise alter them for investigation of specific elements beyond what is there. There are ways around many of these issues, but they are for another video. By no means has this video covered the complete gamut of information that compares the primary and immortal cells. There are specific cases, nuances, and more that are beyond the scope of a YouTube video like this, and they often have industrial experts working on them for years before solutions are found, and well and truly beyond the scope of what we can provide here. The short and distinct differences between them can be seen here in this table, and will provide a full link to the original source, but it summarizes some of the major differences between the primary and immortalized cell lines and how each can be used to its strengths and weaknesses. Thank you for watching this video. If you have found it interesting or useful, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.